How do delta degrees, and biochemical delta differ in Gibbs free energy? Imagine you're watching a chemical reaction unfold. Sometimes it happens on its own, and other times it needs a little push. What determines whether a reaction will happen spontaneously? That's where Gibbs free energy comes into play. It helps us understand if a process is likely to occur naturally or if it needs energy input. Now, within this concept, there are three key terms to know: delta degrees, and biochemical delta. Let's break down what each one means and how they differ. First, delta, or the change in Gibbs free energy, is like the real-time score of a reaction happening in a specific environment. It tells us if the reaction will proceed spontaneously right now. If delta is negative, the reaction tends to move forward on its own. If it's positive, it won't happen without added energy. And if delta is 0, the system is at a state called equilibrium, where nothing changes. This value depends on actual conditions like temperature, pressure, and the concentrations of reactants and products. For example, if you have more reactants than products, delta might be negative, encouraging the reaction to continue. It can be calculated using the formula delta equals delta degrees plus RT times the natural logarithm of Q, where R is the gas constant, T is temperature in Kelvin, and Q is the reaction quotient, which compares current concentrations to those at standard conditions. Next, delta degrees, or the standard Gibbs free energy change, is a fixed number that tells us how favorable a reaction is under ideal standard conditions. These conditions are usually set at 1 atmosphere pressure, 1 molar concentration for solutions, and a specific temperature, often 25 degrees Celsius or 298 Kelvin. This value is like a baseline or reference point. It is calculated from the standard enthalpy change and standard entropy change, using the formula delta degrees equals delta degrees minus T times delta's degrees. This number helps chemists compare different reactions easily, but it doesn't tell us what will happen in a real-world setting, especially inside living organisms. Finally, biochemical delta, sometimes written as delta prime or delta degrees, adjusts the concept of Gibbs free energy to fit the conditions inside living cells. Unlike standard conditions, cells have specific pH levels, ionic strengths, and metabolite concentrations. For example, the pH inside a cell is usually around 7, not the standard 7. Biochemical delta takes these factors into account, making it more relevant for understanding how reactions actually happen in biological systems. It uses the same calculation as delta but with concentrations and conditions that reflected the environment inside a cell. This helps explain why some reactions that seem unfavorable under standard conditions can still occur spontaneously inside living organisms, thanks to the unique cellular environment. So, to sum it up, delta is the current energy change based on real conditions, telling us if a reaction will happen now. Delta degrees is a fixed reference value under ideal conditions, useful for comparing reactions. And biochemical delta adjusts the second one to match the environment inside living cells, giving us a clearer picture of what actually happens in biology. Understanding these differences helps us see why reactions behave differently in labs versus inside our bodies, and how life manages to carry out complex chemical processes efficiently.